Welcome to InformationBoxTicket Lifestyle. My name is Glani, and this is where we explore the latest breakthrough in science and technology. Today, we are diving into a groundbreaking technology that allows researchers to view their mammalian cells in unprecedented detail. Let's start with some basics. First, let's talk about X-rays. Did you know there are two types of X-rays? One is hard X-ray and the second is soft X-rays. Hard X-rays are what we encounter at airport security or if you have ever had a broken bone. They are powerful and they can penetrate deep into materials. On the other hand, soft X-rays are more commonly used in scientific research because they can provide detailed image at a cellular level. Soft X-rays are particularly useful for studying biology, chemistry, and even meteorites. They can reveal chemical information about samples and offer detailed images of structures within cells. However, their use has been limited in live cell imaging due to the damage they cause to the living cells and the specialized equipment required. Let's get to the exciting part. A team of researchers had developed a soft X-ray microscope that allowed them to view mammalian cells for the first time. This groundbreaking tool uses a soft X-ray free electron laser and highly precise Wolter mirrors. Let's break it down how it works. The soft X-ray free electron laser emits ultrafast pulses of light at the speed of femtoseconds. That's quadrillions of a second. These incredible short pulses allow scientists to capture images of cell structure before the soft X-ray radiation can damage them. The other key component is the Wolter mirrors. These mirrors, originally designed for X-ray telescopes, provide a wider field of view and withstand powerful laser radiation without color distortion. This makes them perfect for capturing high-resolution images at various wavelengths. Using this technology, the researchers were able to capture images of carbon-based structures within the living cells, something never seen before with other instruments. Carbon is a fundamental element of life, so this opens up a new window into our understanding of cellular biology. One of the most surprising discovery was the carbon pathway between the nucleolus and the nuclear membrane. The nucleolus is involved in cell function and survival, and this pathway had not been observed with traditional visible light microscopes. The team aims to enhance their microscope by incorporating brighter soft X-ray free electron lasers and even more precise Wolter mirrors. This will enable clear images with less noise and allow the observation of more biochemical elements within cells. Such advancement could illuminate vital reactions and interactions within living cells. In summary, this new technique represents a significant leap forward in our ability to study living cells. By using a soft X-ray free electron laser and Wolter mirrors, researchers can now view detailed images of cell structures without causing damage. This could lead to incredible insight into cellular biology and beyond. Thank you so much for watching till the end. Don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of this Science Find series. Thank you so much and keep watching.